Hello everyone, welcome to MES e-learning channel. So we will continue with the genome editing series of lectures. In the previous two lectures, we have seen two mechanisms, that is your zinc finger nucleases and talons. In this lecture, we would be discussing about Crispr-Cas system. So Crispr-Cas system is the most groundbreaking genome editing tool, and it has transformed the entire molecular biology system. So in this lecture, we will talk about in the nature, what is the role of this Crispr-Cas system. So it is found in bacteria and in archaebacteria, and it is one of the important method of immunity for these your bacteria. So let us understand how it works. So before that, let us see the history of Crispr-Cas. So we can see that around in 1997, it was first discovered that such kind of Crispr-Cas system is present in E. coli. But then, after a lot of time, there was more work that was implicated, where in 2012, first time, it was proposed that this system could be used for genome editing. And afterwards, no looking back, and we have a very good journey of utilizing this particular system for the genome editing. So let us understand that how this particular system gives the adaptive immunity in the bacteria. So what happens is when the viruses attack these bacteria, obviously only the genome is inserted into the cells. So once this genome is inside the cell gets a snippet of these genomes, okay, a small part of this genome, and then it inserts into the between the spacers of this CRISPR loci. So here you can see that these are different colors that is representing the areas, the repeats that are there. So these are called as your spacers. So there are some repeats, and inside them there are spacers. So these spacers are going to be replaced by the snippets of these viruses which are attacking the bacteria. So either it is the viral DNA or the RNA. So it gets attached to it. Once that has been done, then this array, this CRISPR array gets transcribed. So it will transcribe into the precursor CRISPR RNA, and then it will get matured into a proper CRISPR RNA, and this CRISPR RNA is responsible for directing the nuclease, that is your Cas9. Okay, so that's the Cas protein, which is the gene of that is also situated very close to this array of your CRISPR. So together they will guide, so this CRISPR RNA will guide the Cas protein in the corresponding DNA, viral DNA, whatever target is there. So that target is going to be cleaved. Okay, so such kind of arrays are produced and stored in the bacteria, and then later whenever the virus is again attacking this particular bacteria, the bacteria will transcribe it, activate its Cas protein, and then this Cas protein is going to cleave it. It will cleave whatever the viral DNA is there, DNA or RNA is there, it will cleave it and give protection to these bacteria. So this is how the bacteria are going to have this adaptive immune system. Why it is called as adaptive immune system? So adaptive immune system, you already know that this immune system is very precise and is directed towards one type of your pathogen. So here also different types of viruses are going to attack the bacteria. So every time it attacks, such kind of snippets are taken up and then stored in this spacer. It is stored in this CRISPR array. And then later on, whenever there is this viral attack that is there, okay, so what is going to happen is the protection is going to be given to these bacteria if this snippet has already been stored in the array. So this is the mechanism of the adaptive immune system in bacteria. Now let us understand that what are the different types of your CRISPR-Cas systems which are available in the bacteria. So the first one is your type 2 CRISPR-Cas, which is a CRISPR and Cas9 protein combination, which is the most widely used system, and it utilizes one your Cas9 protein and dual RNA system. So here you require two RNAs, one is your CRISPR RNA and the tracer RNA, which together, combined together with your Cas9 will help in recognizing the target sequence and your DNA cleavage. Then you have the type 5 CRISPR, which is a combination of your Cas12a, which is also called as your CPF1 protein, along with the CRISPR RNA. So here it will require a single CrRNA, that is your CRISPR RNA, along with the single Cas protein, that is your Cas12a. And the combination, the characteristics of your Cas12a is different as compared to your Cas9. And then apart from this, we also have your type 1, type 3, type 4 and type 6, which is having different unique characteristics. And as the technology advances, we may also utilize these different types of your Cas9 also in genome editing. At present, the ones that we are using is your type 2 and type 5. So what happens is in the expression and the processing stage, as we have already seen. So once your modification has already been done in the bacterial genome, that is the array has already been created, then the transcription will take place only when the virus is again attacking the bacteria. Okay, so the CRISPR array is transcribed into the long precursor, so that is called as your pre-CrRNA, that is your precursors CRISPR RNA, which contains multiple spacer sequence and repeat sequences. So then you would have the processing it. Okay, so processing in shorter CRISPR RNAs, because every RNA is going to be specific for different types of your sequences, for your target sequences. So each of them would be processed and broken down into shorter CRISPR RNAs, and each containing a single spacer sequence and a portion of the repeat sequences. Then you have the maturation. So the matured cRNA would be now be able to guide. So it will go and bind. Maturation will happen only when your this Cas proteins are going and combining with your Cas proteins with your cRNAs and Cas proteins combining together and going and binding to the target sequences. So once they have bound to the target sequences, then the interference will come. It will cleave the whatever the viral genome is there and bring about the interference in this entire cycle of the viruses. So that's how the entire process will work in the bacteria. So because the system is very flexible, so we can just, there is no combinations or anything, we have to just develop a CRISPR RNA such that it can go and bind to a particular target sequence. This is a very flexible sequence, a very flexible system, and could be used for the genome editing. It is much more efficient as compared to the other two methods that we have already seen, that is your zinc finger nucleases and talents. It is proved to be a lot more efficient, and the success rate is really very high. It is very much versatile and could be used for different purposes, either for your insertions, deletions or modifications, however we want it. And then definitely it has already proved very beneficial for the therapeutic potential. So for different types of cancers, for your infectious diseases, this could be used as a very important genome editing tool in the in vivo systems. So this is what we have seen, that in nature how your CRISPR-Cas system is working. in the next lecture, we will see that how exactly this particular system could be used for the genome editing. Thank you so much, and I hope I could explain you how it is working in the nature. Thank you.